Good afternoon, everybody. Today we will going to discuss about the about second two drugs of alkaloids. In that first drug is a Vinca. Vinca, that is means Sadafoli. Its synonyms Catharyanthus, periwinkle, Sadafoli. Then its biological source. These are dried whole plant of a Catharyanthus roseaceous limb, which is also known as a Vinca rosea, which is known as a Vinca rosea, which is belonging to family Apocynaceae. There is geographical source. Sadafoli, it is indigenous to Madagascar and it is also cultivated in South Africa, India, USA, Europe and in Australia also. Then its morphological characteristics, its color, leaves that is having the green in color. Leaves are green in color, flowers. So Vinca, it is generally found in 3 to 4 colored flowers. That is violet pink color, white color or carmine red in color. Its odor, these are having characteristic odor. Taste is a bitter taste. Then Vinca, it is an erect pubescent herb. It is an erect means saral vadnara, straight vadnara. It is an erect pubescent herb, branched taproot. That is having the branched taproot. Then leaves are simple petiolate leaves. See here, the leaves are simple leaves, then petiolate leaves, ovate, oblong, acute apex, which is having the glossy appearance. Acute apex and which is having glossy appearance. Glossy means chamakdar, which is having glossy appearance or smooth appearance. Then flowers are the complete flowers. See here, flowers that is having 1, 2, 3, 4, 5 numbers of petals. The complete flower means the flower which contains androecium, gynoecium, petals and sepals, which contains sepals, petals, androecium and gynoecium. That flower is called as a complete flowers. Again its fruits. So the fruits are follicles with several black seeds. Fruits are the follicle. See here, see the fruits that are the follicle. Follicle means sheng. Fruits are follicles which is having several black seeds. See here, there are black colored seeds. That is its morphological characteristics. In that color, leaves are generally green in color, flowers are violet or pink colored or white or carmine red colored flowers. Then its odor is a characteristic odor, taste is bitter taste. Then Vinca, it is a pubescent herb, branched root, taproot, and leaves are simple. Petiolate means these are the petiole. Then ovate, oblong, acute, apex and glossy appearance. Okay. After that, its microscopical characteristics. See here the microscopical diagram of the Vinca. In that, first layer is upper epidermis. After transfer section, this diagram is seen under the microscope. That is microscopical diagram or microscopical characteristics. First of all, microscopical characteristics means the characteristics which are seen under microscope, or the characteristics which are studied under microscope. That characteristics are called as a microscopical characteristics. In that, first one is upper epidermis. Firstly, these are divided into two parts, lamina and midrib. So see here, there is a line is observed, that is the midrib, and both sides of lines are the lamina. See here, both sides are lamina. So the microscopic of Vinca, it is divided into two parts, that is lamina as well as the midrib. So here, first of all, lamina. So the microscopical characteristics are the microscopy of Vinca lab, that is the dorsiventral nature. Dorsiventral means upper surface and lower surface that are having similar characteristics, that is the dorsiventral structure. Another is isobilateral structure. Isobilateral means the characteristics of upper epidermis and the characteristics of lower epidermis are not similar. But Vinca drug that is having the similar characteristics. Vinca that is having the similar characteristics that are the dorsiventral nature. In that, first layer is the upper epidermis. These are single layered. See here, upper epidermis, these are single layered rectangular cells with which are covered with a small cuticle. See here, so this lining, this is the upper epidermis, and this upper epidermis which is covered with a smooth cuticle, that which is the smooth cuticle, and this upper epidermis sorry, upper epidermis that contains covering type of trichomes, and a anemocytic type of stomata, that contains covering type of trichomes and anemocytic type of the stomata, that is the upper epidermis. So anemocytic type of stomata means, firstly, stomata means what? It is the minute opening, or it is the small opening which is present at the upper epidermis of leaf or lower epidermis of leaf. It is the minute opening of which is present upper epidermis or lower epidermis of the leaf, and which is used for transpiration of or respiration of the plants. That is the stomata. And this vinca that contains anisocytic type of stomata. Anisocytic type of stomata means it is nothing but unequal celled type of the stomatas. It is also called as unequal celled type of stomatas. In that, 3 subsidiary cells are present. In this type of stomata, 3 subsidiary cells are present. One is smaller than other 2. One stomata is smaller than other 2. That means 3 subsidiary cells are not similar. That's why it is called as unequal celled type of the stomata. Then this is about the upper epidermis. Then second layer is the mesophyll. So mesophyll, which is start from here up to here, means from upper epidermis, below the upper epidermis and above the lower epidermis, the part that is called as the mesophyll. So in mesophyll, first layer is the palisade. So palisade, these are single layered cells. These are single layered elongated cells. See here, these are single layered elongated cells which are compactly arranged to each other. That means which are very compactly arranged to each other. That is the palisade cells. Next one is spongy parenchymatous cells. So the next layer after palisade cells is the spongy parenchymatous cells. So these are generally contains 5 to 8 layered loosely arranged. These contains 5 to 8 layered loosely arranged which are having intracellular spaces. See here, both these 2 cells having intracellular spaces, but these cells are not having intracellular spaces. That is palisade cells, and that is compactly arranged. So there is difference in spongy parenchyma and palisade cells. Palisade cells are very compactly arranged, that are elongated, but the spongy parenchymatous cells and that are single layered, and spongy parenchymatous cells, these are generally 5 to 8 layered loosely arranged intracellular spaces are present, and few cells contains vascular strands. Few cells contains vascular strands, but calcium oxalate crystals are absent. Those candidates who want to appear for GPAT, then note down this point. So the vinca leaves does not present calcium oxalate crystals. So this is about spongy parenchymatous cells. After spongy parenchymatous cells or after mesophyll, the last layer is the lower epidermis. So description of lower epidermis is similar to upper epidermis. So similarly, upper epidermis, as like upper epidermis, lower epidermis are also single layered. That are single layered rectangular cells, and the outer wall of lower epidermis are also present trichomes as well as stomata. But as compared to upper epidermis, lower epidermal cells are contains more number of the stomatas. As compared to upper epidermal cells, lower epidermal cells are having more numbers of the stomata. So this is about the lamina. After that, the next layer is the midrib. Next is the midrib. So here, so this from here up to here, this is the lamina, and this is the midrib. Okay, from here up to here, it is the midrib. So in that, first step is the collenchyma. That is strips of collenchyma. These are the strips. Strips means these are bunch or bands of the collenchyma, strips of collenchyma that are present just below the upper epidermis and just above the lower epidermis. See here, this is also the strips of collenchyma that are present just above the lower epidermis and just below the upper epidermis. Next one are cortical parenchyma. These cells are also loosely arranged as like spongy parenchyma. That is cortical parenchyma. That is cells are also loosely arranged. That is having intracellular spaces. Next one is vascular bundle. So this is the vascular bundles. This is vascular bundle. In that xylem are present and phloem are present. So see here those cells that is having some dot-like cells that are the xylem. Some cells contains dotted that are the xylem. Xylem that are lignified. Lignified means lignin is a present and phloem is a non-lignified, and those cells which are hollow or blankly without any dot, that are the phloem. Okay, so in that xylem are lignified and phloem is non-lignified. After that collenchyma, that is also thick walled and which are present just above the lower epidermis. So that is about the microscopical characteristics of the Vinca. So about this, if you have any problem, then comment me into comment box. Next, so this is the description of microscopical characteristics of Vinca. After that, its chemical constituents. The large number of indole alkaloids which are present into the Vinca. Large number of indole alkaloids, that means indole type of alkaloids are present into the Vinca. Out of them, about 20 dimeric indole and dihydroindole alkaloid possesses. Out of them means so many numbers of indole alkaloids are present in Vinca, but out of them त्यातले फक्त 20 indole alkaloids असे आहेत जे की dimeric indole alkaloids and dihydroindole alkaloids which possesses oncolytic activity. Among of them, most important or two significant, more important are chemical constituents such as vincristine and vinblastine. In that, vinblastine contains indole alkaloid part that is called as catharanthine. Vinblastine contains indole alkaloid. Understand, 20 dimeric indole and dihydroindole alkaloids are present. 20 dimeric indole and dihydroindole alkaloids are present, and that 20 alkaloids that gives oncolytic activity. Oncolytic means which works as like anti-cancerous agents. In that, vincristine and vinblastine are most significant. In that, vinblastine contains indole alkaloid, means vinblastine मध्ये जो indole alkaloid present आहे that part ला म्हणतात catharanthine. That part is called as a catharanthine. And dihydroindole alkaloid, that part is called as a vindoline. Means it contains indole part, that is catharanthine, and dihydroindole part, that is the vindoline. Then along with these, the other alkaloids are also present, that is azmalicin, serpentine, leucinerine and tetrahydroalstonin. Other alkaloids. Continue. So in that, other alkaloids are azmaline, serpentine, leucinerine and tetrahydroalstonin. So it requires about 500 kg crude drug to extract out 1 gram vincristine. It requires means 1 gram vincristine isolate करण्यासाठी approximately 500 kg crude drug is required. It requires about 500 kg crude drugs to extract out 1 gram of vincristine. Its uses: vincristine sulfate, it is an antineoplastic agent which may act by arresting mitosis at the metaphase. Vincristine sulfate, it is an antineoplastic agent. So how it works? It may act by arresting mitosis at the metaphase. Again, it is given intravenously in the treatment of acute leukemia in children. Vincristine sulfate, it is also given intravenously in the treatment of acute leukemia of children. Then in adults, it is used in the treatment of Hodgkin's disease, reticulum cell sarcoma, lymphosarcoma and myosarcoma.